Hey class, in this recording we will focus on antibodies and antigens. So let's dive into it. An antigen is anything, any molecule that can trigger an immune response. And to trigger this immune response, it generally speaking needs to be pretty complicated. The overall molecular mass needs to be at least, as a rule of thumb, 10,000 AMU. And yeah, there's a little fudge factor here. So 10,000-ish AMU. Antigens are typically going to be made of proteins, polysaccharides, glycoproteins, and or glycolipids. And we use antigens all the time in our bodies. We can use them to say, hey, this belongs in our body. This is a self molecule. Or, hey, this is an invader. This is a foreign molecule that does not belong in our bodies. So that's what we use these antigens for. And as we look at these antigens, there are parts of the molecule that we can specifically recognize. So this whole molecule can be an antigen, and the part that our antibodies will specifically attach to is referred to as the epitope, or the antigenic determinant. So it's the part of the antigen that specifically stimulates the immune response. Now, some things are too small to trigger an immune response. Those are referred to as haptens. So it's too small to trigger an immune response all by itself, but if they bind to a carrier molecule, they can trigger an immune response. So when we think of a hapten, here's some generic haptens, and here's the carrier molecule. When the haptens are bound to the carrier molecule, now we have something that can trigger an immune response. We have an antigen. A lot of times, cosmetics, detergents, medicine, poison ivy, animal dander, things people are allergic to are common haptens. By themselves, they don't necessarily trigger an immune response, but when they get into our bodies, they bind to something in our bodies and trigger that immune response. As we think of penicillin, penicillin is a common hapten. It binds to host proteins in the allergic individuals and triggers that immune response. We also have antibodies. Antibodies bind to antigens. And I'm really sorry those terms are so similar sounding. We also could say immunoglobulins bind to the antigens, which is a little bit nicer. It's nice to use different sounding words for different things. So when we think of an immunoglobulin, or a capital I lowercase g, that's another word for antibody. And it's an antibody that we find in our body fluids. These antibodies are all going to be made up of a common subunit, a common monomer that looks like this letter Y on the right side of the screen. This letter Y, or antibody monomer, has 4 polypeptide chains that are going to be held together by disulfide bonds, or bridges. As we look at our antibodies, there's the constant and variable regions. The variable regions are going to bind to specific antigens. The constant region, I'm about to circle the constant in red, this constant region is the same regardless of what antigen the antibody is designed to bind to. So this constant region can be recognized by our body, and then the variable region recognizes the pathogen, or the antigen. That antigen binding site is going to be on that variable region, and it's going to be made of part of the long chain, sometimes referred to as the heavy chain, and part of the short chain, or light chain. And this antigen binding site will attach to the specific epitope of a specific antigen. So as we look at our antibodies, there are 5 broad classes of antibodies we cover in this class. We first are going to cover IgG, the simplest, the smallest, and by far the most common kind of antibody. Immunoglobulin G, IgG, shown here, highlighted in green, is going to be a blood plasma antibody. It's going to be able to also cross the placental boundary and get into the fetus, to get into baby while that fetus is in utero. This IgG, immunoglobulin G, is also going to be secreted as part of the secondary immune response and aids in complement fixation. We also have immunoglobulin A. Immunoglobulin A is a dimer antibody. It's made of 2 monomers, And we find immunoglobulin A in lots of secretions of our bodies, including mucus, saliva, tears, intestinal secretions, and notably, human breast milk. Immunoglobulin A is going to bind to pathogens and can cause multiple pathogens to be bound together. As immunoglobulin A binds to the pathogens that are trying to get through our skin are going to have a more difficult time penetrating the underlying tissues because immunoglobulin A effectively ties multiple pathogens together. It also is going to be responsible for that passive immunity to newborns. I mentioned in a previous recording how newborn children will breastfeed with mom, and they'll take mom's breast milk and get antibodies, get natural passive immunity from mom while mom breastfeeds them. We also have immunoglobulin E. IgE is another monomeric-based antibody. This is an antibody that is a transmembrane antibody, and it's found on basophils and mast cells. This transmembrane antibody means that it's going to be embedded in the cell membrane. And when it's stimulated or activated, it'll stimulate its host cell, the basophil or mast cell, to release histamine and other chemical mediators of the inflammatory response. And if it's an appropriate inflammatory reaction, we refer to that as an allergic response. Immunoglobulin E also is going to attract eosinophils to the site of parasitic infections and is responsible for immediate hypersensitivity reactions. So those very rapid, sudden, and dangerous allergic reactions can be blamed on and are in part responsible or are in part due to immunoglobulin E. And then we have our last ones over here, immunoglobulin M and immunoglobulin D. And as we look at immunoglobulin M, we have the largest one. IgM is a pentamer, and this immunoglobulin M is going to form a rough pentagon of monomeric antibodies, and we find high concentrations of immunoglobulin M in blood plasma and blood lymph. Immunoglobulin M, IgM, is responsible for the agglutination or the clumping that occurs when we have a blood transfusion reaction. It also is capable of triggering complement fixation and is going to be secreted during our primary immune response. So the first time we're exposed to a pathogen, we are going to release immunoglobulin M in response to that exposure. And then finally, we have immunoglobulin D, another monomeric-based antibody. Immunoglobulin D is a beta or B cell or B lymphocyte transmembrane antibody that serves as an antigen receptor on the B cell and helps B cells to be activated when they are exposed to antigens within our bodies. So that's all we have for our antigens and antibodies. If you have any questions or comments, please feel free to post them on the class discussion board or to shoot me an email. Happy studies.